Hello, it's Rudena Nagor, and today we're going to be solving May-June 2018 Variant 2, Paper 1. Some features of cells are listed: cell wall, cell surface membranes, and ribosomes. Which features can be found in plant cells and in prokaryotic cells? For cell walls, yes, both of them are found in both cells. However, in plant cells, the cell wall is made of cellulose, but in prokaryotic cells, the cell wall is made of a polysaccharide called peptidoglycan or myurine. For cell surface membranes, yes, both of them do have a cell surface membrane. For ribosomes, both of them do have ribosomes. However, in eukaryotic cells, both have 70S and 80S ribosomes, but in prokaryotes, it's only 70S ribosomes. Therefore, the answer is A. Which of these statements about light microscopy are correct? The greater the resolution of a light microscope, the greater the detail that can be seen. Of course, this is correct because as the definition of resolution is the ability to distinguish between two separate points. Number 2, the greater the magnification of a light microscope, the greater detail that can be seen. This is not actually correct because it's up to the limit of resolution. Brings us to point 3, increasing the magnification of a light microscope up to its limit of resolutions allow more detail to be seen. That's correct, of course. Number 4, the shorter the wavelength of the light used in a light microscope, the greater the detail that can be seen. This is correct, because coming back to our general rule is that the limit of resolution is half the wavelength used to view it. So the shorter the wavelength, the higher the resolution. Therefore, the answer is B. Question 3. Which type of cell contains the highest proportion of cell structures bound by a single membrane? Here I have some examples of single membrane structures. Example 1 is vacuole, lysosome, Golgi apparatus, and endoplasmic reticulum. Here we have 4 types of cells: it's cellulated epithelial cells, goblet cell, red blood cell, and smooth muscle cell. As we know, goblet cells secrete a lot of mucus, therefore it's normal for them to have a high proportion of Golgi apparatus and endoplasmic reticulum. Therefore, the answer is B. The table shows a variety of structures which may be found in cells. Which row shows the structures that could be found in root cells of a plant? So it's a plant cell. To start with, plasmodesmata. Yes, of course, plant cells have plasmodesmata because according to our definition, plasmodesmata are strands of cytoplasm projecting from cell walls. So this is correct. To the second point is glycogen granules. Glycogen granules is the storage polysaccharide in animal cells only. Therefore, it's not in plant cells. So this is correct. Golgi body. Now, all eukaryotic cells do have a Golgi body, therefore it's correct. And plant cells also do have mitochondria, therefore it's also correct. Therefore, the answer is B. Question 5. Which of these cell structures are present in Plasmodium? To start with, we all know that Plasmodium is the organism that causes malaria. We all know malaria is a eukaryotic cell. Therefore, eukaryotic cells do have ribosomes, mitochondria, and Golgi body. Therefore, the answer is A. Question 6. A student was asked to estimate the concentration of glucose in a solution using a Benedict's test. The student was provided with a 1 mol dm cubed glucose solution and was told to make a 0.6 mol dm cubed solution by proportional dilution. Which row shows the correct volumes of both 1 mol dm cubed glucose solution and distilled water needed to make the 0.6 mol dm solution? First of all, we divide the volume distilled by water by the volume of the glucose solution. And the volume that gives us 0.6 mol dm is going to be the correct answer. In this case, 8 divided by 12 is going to give us 0.6. Therefore, the answer is A. A diagram shows part of a carbohydrate molecule formed by glucose. What is the name of the molecule? Here you could see a highly branched chain. In this case, we know it's going to have 1,6 glycosidic bonds. Therefore, let's see the suggestions. Here we have amylose. It's an unbranched molecule and it's only 1 to 4 glycosidic bonds. Therefore, it cannot be amylose. Cellulose. Cellulose is not branched, so this is also incorrect. Glycogen contains 1,4 glycosidic bonds and 1 to 6 glycosidic bonds, and it is branched. Therefore, might be correct. D, we have starch. Starch contains both amylose and amylopectin. As we know here, amylose is not branched, therefore it cannot be starch, therefore the answer is C. Question 8. The diagram shows a relationship between some biological molecules. Which row is correct? Let's start with A. Here is alpha glucose, carbohydrate, and sucrose. 3 of these are correct. However, here it's assuming that sucrose is a monomer. However, this is incorrect because sucrose is made of alpha glucose. It's a disaccharide made of alpha glucose and beta fructose. Therefore, it's not a monomer. So this is incorrect. For B, 1 is cellulose, polymer, starch. These are correct so far. Starch is a carbohydrate, so this is correct. However, here it's amylase. Amylase is a protein. It's not a carbohydrate. Therefore, this is incorrect. For C, we have fructose, reducing sugar. This is correct. Fructose is a reducing sugar. We have beta glucose. It is also a reducing sugar. And it is a monomer, and amino acids are monomers. So this might be correct. For D, we have hemoglobin, protein, amylose. However, amylose is not a protein, therefore this is incorrect. Therefore, the answer is going to be C. Question 9. Which diagram shows the reaction that occurs to join two monomers that form cellulose? As we know, cellulose monomers are beta glucose molecules and joined by beta 1 to 4 glycosidic bonds. To start with A, here it's assuming that this is alpha glucose and this is also alpha glucose. However, this is incorrect because, as I said, cellulose is made of beta glucose monomers. For B, the second monomer is not rotated, so this cannot be correct. For C, the same issue, it's also alpha glucose and alpha glucose. For D, this is beta glucose, so this is right. And the second monomer is rotated 180 degrees, so that is also right. And the glycosidic bonds forms perfectly here. Therefore, the correct answer is going to be D. 10. How many molecules contain C double bond O bonds? For amino acids, let's just draw out an amino acid. So here, you can see a carbonyl group. Therefore, this is correct. For fatty acids, because here it says acids, then it also contains or must contain a C double bond O group. Therefore, it is correct. For glycerol, it's an alcohol, so it does not contain a carbonyl group. Therefore, the correct answer is B. Question 11. Which row describes a collagen molecule? To start with, I've written out the structure of collagen. First of all, it's fibrous, a fibrous protein being insoluble and not metabolically active. Number 2, it's a triple helix made up of 3 polypeptide chains. Every third amino acid is glycine, therefore they coil really tightly. Number 4, H bonding between polypeptides. Yes, there are hydrogen bondings between the 3 polypeptides, therefore it has a high tensile strength. Number 5, covalent and some hydrogen bonding between the molecules. When we form the molecules with 3 polypeptide chains, these join together by some covalent bonds and some hydrogen bonds to maintain the high tensile strength. Coming back to our questions, let's start with A. The bond stabilizing the molecule. We should know by now that it's definitely not disulfide, so this is incorrect, and hydrogen is correct. Properties. High temperatures increase flexibility. We all know this is incorrect. Properties. High tensile strength. Yes, that's correct. Insoluble in water, also that's correct. Primary structure. Repeat sequences of 3 amino acids, this is correct. High proportion of the glycine and proline. Yes, these are correct. These are the amino acids that form the polypeptide chains. For the helix, here it's triple helix. Yes, it's correct. We mentioned that earlier. But it's definitely not alpha helix because alpha helix is a secondary structure and we do not have secondary structure here. Therefore, the answer is going to be C. A student investigated the hydrolysis of the lipid in high-fat milk using enzyme lipase. 1 centimeter cubed of enzyme solution was added to 10 centimeter cubed of high-fat milk. The temperature was kept constant. The pH of the reaction mixture was recorded at time 0 minutes and every minute for 20 minutes. Which statements could be supported by the results of the investigation? Less product is made as time proceeds because the substrate is decreasing. This is normal because as the number of enzyme substrate complexes increase, the product also increases, therefore the substrate decreases as the reaction goes on or proceeds. Number 2, the pH of the reaction mixture changes more rapidly in the first few minutes and then changes less rapidly. Why? Because at the start the rate of the reaction is really high because there is a lot of substrate molecules and there is a lot of enzyme substrate complexes. Therefore, there's a higher rate of reaction at the start. Therefore, the pH changes more rapidly. Fats are hydrolyzed to fatty acids and glycerol. We all know that fatty acids have a low pH, therefore the pH reaction changes more rapidly. And this is correct. The product gradually causes more lipase molecules to denature. Yes, that is correct because as the pH decreases, the hydrogen and ionic bonding is altered due to the increased number of H+ ions in the mixture. Therefore, the tertiary structure changes shape and becomes no longer complementary. Therefore, this is correct. Therefore, the answer is going to be A. A student carried out an investigation into the effect of temperature on the rate of an enzyme catalyzed reaction. At each temperature, the substrate concentration was measured after 10 minutes. All other variables were kept constant. Which graph shows the effect of increasing temperature on the substrate concentration after 10 minutes? Okay, the first point we have here is at each temperature. So the whole graph represents different experiments at different temperature. Take that into account. This is really important. So what we have here, we obviously know at the optimum temperature, the substrate concentration decreases the most because there are the most enzyme substrate complexes being formed at this time. Therefore, it's obvious that the substrate concentration decreases rapidly at that optimum temperature. And by the way, look carefully at the axes. It says here it's substrate concentration, not the rate of reaction. As we start changing the temperature or increasing the temperature, the kinetic energy of molecules increase. Therefore, there are more collisions with the enzyme, with the substrate. Therefore, there's an increase in the rate of reaction. And as the temperature continues to increase, the active site molecule starts to vibrate rapidly and bonds are starting to break. The first bonds to break at higher temperatures are definitely hydrogen bonds because they are the weakest bonds. Therefore, the shape of the active site is started to get altered, okay? Until the active site is no longer complementary to substrate, therefore no more substrate is used up. Therefore, the answer is going to be D. Question 14. The graph shows how the rate of an enzyme catalyzed reaction depends on the concentration of the substrate. What is the Michaelis-Menten constant for this enzyme under these conditions? So, for the Michaelis-Menten constant, it's the substrate concentration at half Vmax. So first of all, we have to work out what the Vmax is. The Vmax is where the plateau lies. And we have to work out half Vmax. Half Vmax is going to be here. Then we all know that Michaelis-Menten constant is the substrate concentration at half Vmax. Therefore, we read off the concentration and it's going to give us 1.5. Therefore, the answer is going to be C. Question 15. The formula shows how the rate of diffusion across the cell surface membrane can be calculated. Which row shows how the fastest rate of diffusion can be achieved? So we all know increasing the surface area increases the rate of diffusion. Therefore, high surface area. Difference in concentration. We all know that the higher difference in concentration gradient, the greater the diffusion. Therefore, these are also correct. The thickness of the membrane. Having a low membrane thickness means the distance that they have to travel is less and the time that they take in to travel is also less. Therefore, having a low thickness of membrane is going to have us achieve the fastest rate of diffusion. Therefore, the answer is going to be B. The diagram shows part of the cell surface membrane of an active animal cell. Which statement correctly describes the net movement of molecules across this membrane? The first point, oxygen diffuses through molecules M from X to Y. To start with, this side is the outside of the cell, and this side is the inside of the cell. Then how did you distinguish that? We could clearly here see glycoproteins and glycolipids on the membrane, and these are always to the outside because they are hydrophilic and they form hydrogen bonds with the exterior fluid of the cell. Now coming back to our question, which statements correctly describe the net movement of molecules across this membrane? The first point is that oxygen diffuses through molecules M from X to Y. This is correct because oxygen is a non-polar molecule and can diffuse through the hydrophobic core of the cell surface membrane. So this is correct. Carbon dioxide diffuses through molecules M from X to Y. From X to Y is totally incorrect because carbon dioxide does not diffuse from outside the cell into the inside of the cell. So this is totally incorrect. Water moves from Y to X through molecule L. To start with, a more negative water potential means a lower water potential. And a less negative water potential means a higher water potential. For water moves from higher water potential to lower by osmosis from Y to X. This is correct. Through molecule L, or requires a hydrophilic carrier protein to enter the cell. Therefore, the answer is going to be C. Some epidermal cells from a well-watered plant are placed in 3 solutions which have different water potentials. Which row correctly shows the state of the plant cells in each of these solutions? Water potential of the solution lower than the cell. Let's just draw the cell out first. Water potential of the solution lower than the cell. If the water potential of the solution is lower than the cell, then the cell has higher water potential. Therefore, water goes out of the cell down water potential gradient by osmosis. Therefore, the cell becomes plasmolysed. When it's equal to cells, it's turgid because there is no net movement of water inside or outside the cell. If the water potential of the solution is higher than the cell, then water moves in to the cell by osmosis down water potential gradient. But because plant cells have a strong cellulose cell wall, then they are able to withstand turgor pressure and they do not burst, unlike animal cells. Therefore, the correct answer is turgid, and it's B. Question 18. One of the chromosomes in a nucleus has a telomere which contains many repeats of the base sequence. This chromosome was extracted from 4 different cells and the total number of bases in the telomere were determined. Which total number of bases was found in the cell that has undergone the most mitotic division? So there's a general rule that we have to know about. The cell which has undergone the most mitotic divisions has the least number of telomeres. Why? Because each time the cell divides, a part of the telomere is lost. and if it divides again, another part of the telomere is also lost. Therefore, the cell which has undergone the most mitotic division has the lowest number of telomeres. In this case, the answer is going to be A. The chromosome content of each daughter cell must be identical to that of the mother cell for a successful cell replication when repairing tissues. Which stages of mitosis make sure that each daughter cell receives one chromatid from each chromatid pair? To start with, there are some definitions that we have to know. In metaphase, the chromosomes align along the metaphase plate. Spindle fibers attach to the centromeres of the two sister chromatids. They attach right here. During anaphase, the spindle fiber shortens and pull the two sister chromatids apart to the opposite poles. Totally like this. Therefore, the stages of mitosis that make sure that each daughter cell receives one chromatid from each chromatid pair must be metaphase and anaphase. Therefore, the answer is B. Number 20. Which of these statements about cytokinesis is always true? The cell structures replicate. No, they do not replicate. They replicate in the G2 phase. Number 2. Cell structures are shared between two cells. This is correct, but not necessarily shared equally. Number 3. Nuclear envelope reforms. This is incorrect because the nuclear envelope reforms during telophase, not cytokinesis. Therefore, the answer is going to be C. 21. Which statement is correct about the number of telomeres present in prophase of a human body cell? To start with, we have to know where are telomeres in the first place. Therefore, drawing out the chromosome, there are telomeres in each chromatid. So there are 2 telomeres on each chromatid. And this is a single chromatid. Therefore, the correct answer is going to be D. As here it mentions that there are going to be 124 as there is one telomere at each end of the 92 chromatids. Therefore, D is correct. Question number 22. Which statement explains why DNA replication is described as semi-conservative? To start with, half of each original strand is conserved in each new molecule of DNA. No half strands are conserved at all, so this is incorrect. Half of the base sequence of each strand is conserved in the new molecule of DNA. This is also incorrect because no half strands are conserved. C. Only one strand of DNA is used as a template during replication. This is incorrect because both strands, the one lying from 3' to 5' is used, and the one from 5' to 3' is also used. D. The template for each new molecule of DNA is the one strand of the original molecule. That is correct. Question number 23. A section of DNA molecule has the coding sequence. Which statements are correct? This coding sequence contains 3 pyrimidines. This is correct because pyrimidines are cytosine and thymine. Therefore, here we could see that there are 3 pyrimidines. Therefore, this is correct. The double-stranded DNA of this section contains 26 hydrogen bonds between the complementary bases. This is correct because there is a general rule that we have to know. For cytosine binding with guanine, it forms 3 hydrogen bonds between them. But for adenine binding with thymine, it forms 2 hydrogen bonds. If you count the number of bonds formed, you're going to find it's going to be 26 hydrogen bonds. Therefore, it is correct. Question number 3. The double-stranded DNA of this section has 20 sugar phosphate bonds. This is incorrect because if we count here, 1, 2, 3, 4, 5, 6, 7, 8, 9, and the other strand is also going to have 9, therefore it's going to be 18, not 20. Therefore, this is incorrect. Therefore, the answer is going to be B. XNA is a laboratory-made nucleic acid made of nucleotides in which one component has been replaced by a chemical X. The chemical X is not found in nature. The part of the molecule responsible for coding is not changed. Which organic component of DNA or RNA nucleotide has been replaced by X? So as we know, DNA or the X or the D, what it represented is the deoxyribose sugar, and it's the pentose sugar right here. Then we know that pentose sugar is also a 5-carbon sugar, therefore the answer is going to be A. And a side note, for DNA the sugar is deoxyribose, but for RNA the sugar is only ribose. Question number 25. Which statements about tRNA are correct? Hydrogen bonds between bases temporarily hold tRNA against mRNA. This is correct when tRNA binds with the RNA, the anticodon of the tRNA molecule binds with the codons on the mRNA molecule. Therefore, this is correct. Question number 2. The base sequence in the tRNA molecules are the same as the base sequence in the mRNA that is being translated. This is incorrect because the base sequences in the tRNA molecules are complementary to the DNA, however, it has uracil instead of T. So this is incorrect. Point number 3, the specificity of the tRNA molecule for glycine and the specificity for the enzyme that loads glycine are both necessary for the correct loading. This is correct. Therefore, the answer is going to be C. Question number 6, what is the role of ATP in a companion cell when sucrose is loaded into a sieve tube element? Let's quickly describe the process of the loading of sucrose into a companion cell. First of all, the companion cell starts out by pumping H+ ions outside to its cell wall by proton pumps. Then takes one sucrose molecule and goes back into the companion cell via a co-transporter protein by facilitated diffusion. That's like a general explanation. Moving sucrose into a sieve tube element. Definitely ATP is not directly needed to move sucrose into a sieve tube element. B. Removing protons from the sieve tube element. Protons are not removed from the sieve tube element, it's removed from the companion cell, therefore this is incorrect. C. Removing protons out of the cytoplasm of the companion cell. This is correct. D. Taking up sucrose into the cytoplasm of companion cell. This is also incorrect. Therefore, the answer is going to be C. Question 27. A potometer was used to measure the transpiration rate of leafy shoot. Let's quickly explain what is transpiration. Transpiration is loss of water vapor through the stomata. What could be the correct units? The correct unit is always going to be D, centimeter cubed per minute. Question 28. Which combination of features is characteristic for a Phloem sieve tube element as it is loaded from a source? So as a Phloem sieve tube element is loaded from a source, the water potential definitely decreases because now there are a lot more of sucrose solute molecules inside the Phloem sieve tube elements at the source. Okay? For the lignification of cell walls, definitely Phloem sieve tubes are not lignified because there are living cells. 29. The diagram shows transverse sections of parts of a plant. Which structures labeled 1 to 4 transport water and sucrose? What transports water and sucrose is definitely the Phloem sieve tube elements. Therefore, in a leaf, the xylem is at the upwards and the phloem is downwards. Therefore, 2 is the phloem, and in a root, 4, or the cross sign, is the xylem, and these edges are definitely the phloem. Therefore, the answer is C. Question 30. Which statement explains why the ventricles contract after the atria? To start with, the band of fibers between the atria and the ventricles conducts the excitation wave slowly. This is not possible because these fibers are actually non-conducting fibers. So these never conduct the excitation wave. For B, the excitation wave has to pass through a small area of the conducting fibers into the septum. And here it's talking about the AVN node. And the role of the AVN is it delays the impulse and relays it. However, it delays it 0.1 seconds to give a chance for the atria to contract first before the ventricle. So this is totally correct. For C, the excitation wave has to travel further to reach the base of the ventricles than to spread across the atria. This is not correct. The excitation wave travels slowly through the Purkinje tissue to reach the base of the septum. This is also incorrect. Therefore, the answer is going to be B. Question 31. If the left atrioventricular valve in the heart does not close completely, it results in blood flowing back into the atrium during ventricular systole. Which would be the immediate effect of this backflow? 1. Blood flowing from the heart carries less oxygen because less blood enters the lungs. This is incorrect because the left side, here it says the left, it's talking about the oxygenated blood. Here I'm going to give you an acronym that will really help you, and it's LORD. This stands for left is for oxygenated and right is for deoxygenated. Pressure in the left ventricle falls because less blood enters the pulmonary artery. Also, it's incorrect because the pulmonary artery is from the right ventricle, not the left. C. The lower systolic pressure in the left atrium and less blood enters the pulmonary artery. This is also incorrect because of what we just talked about. The raised pressure in the left atrium and less blood enters the aorta. Raised pressure in the left atrium because now the left atrium has to contract more blood into the ventricle, so it generates more pressure. And because the blood, instead of going to the aorta, now goes back into the atrium, so less blood enters the aorta. Therefore, this is totally correct. The answer is D. Question 32. The graph shows two oxygen dissociation curves, one for tissue at rest and one for the same tissue during exercise. What factors have caused the difference in the position of the dissociation curve during exercise compared with the dissociation curve for the tissue at rest? To start with, tissue during exercise has an increased concentration of carbon dioxide. This means that more carbon dioxide and water are going to be converted into carbonic acid by the enzyme carbonic anhydrase. Therefore, this carbonic acid dissociates into H+ ions, which stays inside the red blood cell, and HCO3 ions, which actually leave the red blood cell. Therefore, number 1, an increase in pH. This is incorrect because an increased number of H+ ions means a decrease in pH. So the second point is correct. Point 3, increase in hemoglobin affinity for O2. This is incorrect because as H+ ions binds to hemoglobin, it forms hemoglobinic acid, which has a low affinity for oxygen. Therefore, hemoglobinic acid unloads oxygen actually faster than the normal hemoglobin. Therefore, there's a decrease in hemoglobin affinity for O2. Therefore, 2 and 4 is correct, so the answer is D. Question number 33. Which of these statements about the formation of hemoglobinic acid are correct? Number 1. It can only occur with the dissociation of oxygen from hemoglobin. This is correct because hemoglobinic acid occurs because of the formation of H+ ions. When H+ ions binds to hemoglobin, now hemoglobin has a much lower affinity for oxygen and then it releases oxygen. Therefore, the correct answer is A. Number 2, it removes excess hydrogen ions by preventing blood coming too acidic. This is correct because H+ ions binds to hemoglobin, which removes it from the blood. This is also correct. Number 3, it is linked to the action of carbonic anhydrase. Also, we mentioned up that carbonic anhydrase converts carbon dioxide and water into carbonic acid, which dissociates into H+ ions and bicarbonate ions. Therefore, the answer is A. Question number 34. Which of these structures contain cartilage and cilia? First of all, cartilage, the only structures that contain cartilage are bronchi and trachea. Bronchi contain irregular blocks of cartilage. Meanwhile, trachea contain C-shaped cartilage. So the answer is going to be C. 35. What correctly describes the effect of carcinogens on lung tissue that causes a tumor to develop? We all know that a tumor develops due to a mutation or a sudden change of base sequence of DNA. Point C mentions that DNA changes, causing bronchial epithelial cells to divide by mitosis in an uncontrolled way. Here, when a mutation occurs, the proto-oncogene turns into an oncogene, which therefore causes the uncontrolled division. Therefore, the answer is C. 36. Which row identifies both the type of pathogen that causes cholera and the way in which cholera is transmitted from person to person? First of all, the pathogen that causes cholera is Vibrio cholerae, and it's transmitted by fecal, oral, root, or contaminated water. First of all, the type of pathogen is definitely a bacterium. The method of transmission is drinking water contaminated with the pathogen. That's totally correct. Therefore, the answer is A. Question number 37. Why do people with HIV/AIDS have a higher occurrence of malaria than people without HIV/AIDS? First of all, HIV is human immunodeficiency virus, and it infects T cells and suppresses the immune system. To start with, HIV/AIDS and malaria are both diseases where the pathogen travels in blood. This is actually correct, however, it's not addressing the question. Therefore, it's incorrect. B. HIV/AIDS and malaria are both transmitted in the blood. This is correct, but it does not address the question too. HIV/AIDS infects T lymphocytes, which are used by the malaria pathogen. It's not used, so this is incorrect. HIV/AIDS suppress the immune system. This is totally correct. Question number 38. How many variable regions are there on a typical Y-shaped antibody molecule? There are 4 variable regions. 2 on the light polypeptide chains and 2 on the heavy polypeptide chains, or the longer ones. Therefore, the answer is going to be C. Where are antigens found? On the surface of pathogen, yes, they are found on surface of pathogens, the non-self antigens, and on the surface of macrophage. This is correct because macrophage, once it has engulfed the pathogen, it becomes an antigen presenting cell presenting that same antigen. Therefore, the answer is A. Question number 40. When an organ is transplanted from one person to another, rejection of the non-self organ must be avoided. At the same time, the immune system of the recipient must be maintained to prevent death from the infections. What prevents rejection of the transplanted organ? Continued activity of B lymphocytes. This is incorrect. Natural active immunity, no. Natural passive immunity, also no. Suppression of T lymphocyte activity, this is correct. Therefore, the answer is D. Thank you very much for watching, and please subscribe and share this video.